Curious facts about the Egyptian mummies. Welcome to our new video. In this video, I'll present some curiosities about Egyptian mummies. Get ready to learn surprising things about this part of Egypt's history and amaze your friends with these interesting facts. Mummification in ancient Egypt aimed to prevent the natural process of decay. Mummification was crucial because of its religious significance, as the belief in the afterlife in Egyptian culture was linked to the preservation of the body. It consisted of a complex funerary ritual involving the gods Anubis, Nephthys, Aset, and Osiris. Death marked the beginning of a new cycle and the separation between the soul, Ba, and the body, Ka. Initially, the most used techniques were linen wraps or resin or natron shrouds. The legend of Osiris had a significant influence on the development of embalming techniques. In this legend, Anubis resurrects Osiris with the help of magical formulas. Hence, Osiris represents not only death, but also the rebirth of life, while Anubis is considered the patron of embalmers. Organs were extracted from the bodies, cleansed, embalmed, wrapped in linen cloths, and placed in 4 canopic jars representing the sons of Horus and protecting the organs: Amset, vessel with a human head guarding the liver; Hapi, vessel with a baboon head guarding the lungs; Kabesanuf, vessel with a falcon head, where the intestines were deposited; and Duamutef, vessel with a jackal head guarding the stomach. The heart was usually left inside the body because it symbolized the individuality of each person, being the seat of thought, feelings, life, and consciousness. Mummification reached its peak in the New Kingdom, 1550 BC, 1070 BC, managing to maintain the expression of the deceased's face. Examples of mummies from this period include Seti I and Ramses II. Did you know that the mummification process lasted for 70 days? The corpse was washed, the viscera removed, then the body was left to dry in the sun and covered with several layers of oils, ointments, aromatic oils, and conifer resin. In addition to the oils, beeswax was also applied to the body, which, thanks to its antibacterial and hydrophobic properties, played an important role in the embalming process. Once the previous stages were completed, the bandages were applied. Among the different layers of bandages wrapping the body, protective amulets were placed. In some cases, a network of ceramic beads was also applied. The body of the deceased was then placed in 1 or more sarcophagi. Sometimes a gold or painted mask covering the face of the deceased was placed. There were several embalming techniques, some more costly than others. During the Old and Middle Kingdoms, mummification was exclusively for members of the pharaoh's family and officials and courtiers whom the pharaoh granted this privilege. As mummification began to spread, workshops dedicated to embalming emerged. During this ritual, several people were involved, with the most prominent being the Lord of the Secrets, who wore an Anubis mask and was responsible for treating the deceased's head. The priestly readers would recite the ritual instructions and magical incantations. The cutters were responsible for making incisions and extracting the viscera from the corpses. A curious and somewhat peculiar fact is that mummy extract was prescribed as medicine during the Middle Ages. The name of this exquisite medicine was mumia, and it was indicated for the treatment of headaches, bubonic plague, and indigestion. It is thought that this medicine may have arisen from a mistranslation of Persian or Arabic texts. Different forms of mummy extract were found, often represented by the liquid released by dead bodies mixed with aloes, pieces of mummy flesh, or a spice found in the tombs of the dead. Initially, powdered human remains, resins, and pieces of mummies sold in European pharmacies were imported from Egypt, but even though authorities prohibited such behavior, trading with local remains continued. Thank you for watching this video. If you liked it, don't forget to like and subscribe to my channel for more content like this. And tell me in the comments, what do you think of mummy extract? How does
Curiosidades
cloning
sobre el
work?
clonaje.
Have humans already been cloned? Welcome to our new video. In this video, I will present to you some curiosities about cloning. The following topics will be addressed: what is cloning? How does animal cloning work? Types of artificial cloning, successfully cloned animals. How does therapeutic cloning work? Has any human been cloned? The advantages and disadvantages of cloning, and ethical questions about cloning. Get ready to learn amazing things about this procedure and amaze your friends with these interesting facts. What is cloning? The term encompasses various techniques aimed at generating a genetically identical copy of a biological entity. The first thing that comes to mind is the cloning of entire organisms, but genes, organs, and tissues can also be cloned. For the cloning of an extinct species that still has similar animals, an intact cell could be used, or the entire genome of the extinct animal would need to be mapped to reconstruct its genome, and the genome of the existing animal modified to transform it into that of the extinct animal. The ovum would then be implanted into a similar animal until the species had enough individuals to continue existing on its own. Studies have shown that both the meat and milk of cloned animals are not toxic to our bodies and generally do not show differences when compared to the same natural products. Clones are not only artificially generated in laboratories, but asexual reproduction also exists in nature, generating a new identical individual from a single cell of the parent. In mammals, something similar can be observed with identical twins. It's worth noting that in mammals, the DNA of the offspring is different from that of the parents, and even twins do not share the same DNA 100%. Types of artificial cloning. There are 3 types of artificial cloning: genetic cloning, reproductive cloning, and therapeutic cloning. The difference between them lies in the outcome of the process. Genetic cloning produces copies of DNA segments or genes. Reproductive cloning produces entire individuals. And therapeutic cloning produces embryonic stem cells for tissue creation and reconstruction. How does genetic cloning work? The gene of an organism is inserted into a vector, bacteria, viruses, yeast cells, or plasmids, and then the necessary conditions are provided for the vector to multiply. How does animal cloning work? In reproductive cloning, a mature somatic cell of the organism to be cloned is needed. This sample will be inserted into an empty ovum or oocyte, meaning this cell had its own DNA removed and is now nucleus-free. The insertion of the cell's DNA into the ovum can be carried out in 2 ways. The first is to fuse the entire cells using an electric current. The second is to extract the nucleus of the somatic cell with a needle and inject it into the ovum. Initially, the first stages of embryo development occur in a test tube, and then the embryo is implanted into the womb of an adult female animal. This technique is called somatic cell nuclear transfer. The main challenge for these animals is to survive birth. Those that survive are usually healthy. This technique has made it possible to increase the success rate and therefore reduce the mortality rate. Successfully cloned animals. The most famous case of successful cloning is that of the sheep named Dolly in 1996. It took 277 cloned embryos to create her. But did you know that Dolly wasn't the first successfully cloned animal? In 1979, the first cloned mice were produced, and then cows, chickens, and sheep were cloned. And now you might be wondering why Dolly is the most famous cloned animal? The cloning of this sheep was different from the previous ones. It was the first time that a mature somatic cell was successfully cloned. The previous cases involved injecting embryonic cells into an empty ovum. The San Diego Zoo maintains DNA samples from endangered animals. Using one of these samples, 2 banteng, a bovine native to Southeast Asia, were cloned. One of the animals, Noha, had an infection, and the other, Jahava, had to be euthanized. African wildcats have been successfully cloned using frozen embryos as a source of DNA. The first successfully cloned pet was a Maine Coon cat named Little Nicky, which honors its predecessor Nicky. The owner of the cat spent $50,000 on this procedure, which was carried out by Genetic Savings and Clone. This company had already cloned the first cat in 2001. How does therapeutic cloning work? They create a cloned embryo to produce embryonic stem cells with the same DNA as the original donor cell. This process helps to learn more about diseases and to treat them. The stem cells are removed from the embryo when it is still in the test tube during the blastocyst phase. At this stage, there are approximately 100 stem cells that make up the embryo. Further studies are needed to understand stem cells and cancer cells, as both have the ability to proliferate indefinitely. There are even studies that have shown that after 60 cycles of cell division, a stem cell can become a cancer cell. Has any human been cloned? Some studies and institutions have claimed to have achieved it, but they have never presented evidence of it. Cloning of humans and primates is more challenging than with other mammals because the correct cell division requires 2 proteins called fusiform. These proteins are located very close to the nucleus in human eggs, and during the process of removing the nucleus from the egg, they can be damaged. Other mammals have this protein located throughout the egg. Scientific applications for cloned animals. Scientists who cloned Dolly conducted a study to alter the milk production of sheep and add a protein for blood clotting. The goal of this study is to purify this protein in the future and provide it to people with blood clotting deficiencies. That's why these modified sheep were cloned. Another significant contribution of cloned animals to science is that, being genetically identical, they should react in the same way to drugs. This would allow for more effective preclinical drug testing. Advantages of cloning. DNA from prize-winning bulls that can be valuable to farmers can be preserved. Animals with better milk or lean meat production or other desirable characteristics can be cloned. Recover lost animal diversity over the years. This would allow for interaction with these specimens, research, and observation. We can already see endangered animals in several zoos, but in the future, there could even be zoos with animals that are currently extinct. Being able to appreciate these incredible animals that ended up extinct due to climate or hunting reasons. Disadvantages of cloning. The main challenge when cloning animals is the cost of the procedure. When it comes to extinct animals, this cost increases due to the difficulty of finding specimens that can be used as evidence. As they are cloned from adult cells, the telomeres of the chromosomes are shortened. This is normal as organisms age, but cloned animals have shorter chromosomes compared to normal individuals of the same age. These animals still have a high mortality rate. It is thought that this is due to a resistance of the donor egg that prioritizes genetic memory over replaced genes. They can also present defects in vital organs. Due to the high mortality rate, there is an ethical problem of bringing animals to life that may die, without knowing if they will be healthy or not. Some of the animals that became extinct a long time ago would not adapt to the current condition of our planet. A way to not only replicate but also evolve their genome or provide a more suitable habitat would be needed. Cloning produces genetically identical individuals, so it's not possible to maintain the necessary diversity for species survival. Cloned animals, even with the same genetics as the original animal, may have different phenotypic characteristics. In some species, genes responsible for coat color are randomly activated and deactivated. Environmental conditions also influence animal appearance. Cloned animals can be born larger than normal. They can age prematurely and have immune problems. Ethical questions about cloning. Given the high mortality rate, many question whether it is ethical to bring animals to life that do not survive the experiment or even have health problems. Others defend this practice because if the procedure is never done, it cannot be perfected, and therefore this problem cannot be solved. Some see cloning as an opportunity to reduce costs for future gastronomic or hunting experiences. But there is also the problem that animals are living beings, and raising animals in laboratories with the sole purpose of killing them does not seem ethical. On the other hand, it would solve the problem of overfishing and the risk of extinction of some of these specimens. Many question the practice of breeding these animals for tourism and keeping them in captivity because it would not allow them to be in their natural habitat or live freely. If the DNA of these extinct animals is combined with that of their current relatives, will the cloned animals truly be the return of the extinct animals or simply a variation of them? Some scientists believe that if the missing genes are not exclusive characteristics of this species, they could be brought back. Could these animals have a negative impact on our planet's ecology? Conclusion. Cloning will continue to evolve, starting from current specimens, until it becomes possible to clone animals that recently went extinct, and then evolving to clone animals that have been extinct for longer periods. Currently, only DNA up to 1 million years old can be cloned, so animals that lived longer ago cannot be cloned. Cloning could bring these animals back and preserve diversity, as long as animals are treated with respect and provided with dignified conditions. This practice, along with current animal preservation measures, could help many species. Bringing back extinct animals is possible, but we are still far from achieving this due to the difficulty not only of cloning these animals but also of bringing the necessary diversity for them to mate and produce healthy individuals with biological diversity that do not share the exact same genetic code. Thank you for watching this video. If you liked it, don't forget to give it a like and subscribe to my channel to receive more content like this. And tell me in the comments, what do you think about cloning? Lately, there has been a lot of talk about infinite energy and perpetual motion machines, but is this really possible? In this video, I will tell you the myths and truths about these concepts. Energy can be measured in joules, calories, and kilowatt-hours, and it has the ability to do work. This energy can manifest in various forms, such as mechanical, nuclear, electromagnetic, and thermal energy. Energy cannot be created or destroyed, it can only change its form. There are two types of perpetual motion machines. The first type aims to produce more energy than it consumes, and the second would perform work by exchanging heat with only a single thermal source. The problem with these research paths is that they violate the first and second laws of thermodynamics. Many of these machines use asymmetry combined with gravity to make the motion repeat over and over. However, to start these machines, an initial push is needed, which, with the help of asymmetry and gravity, manages to maintain the motion for a certain period of time, but the energy gradually dissipates. Although this idea has become very popular recently, its origins date back to the 12th century with Bosquer's wheel. This wheel relies on the displacement of fluids by gravity to keep the wheel in motion. Throughout history, various machines have been invented that continued to function with the intent of achieving a perpetual motion machine. Although these machines continued to perform their tasks for a certain period of time, they eventually stopped working. And what is the archenemy of infinite energy? Friction. The motion continuously loses energy until there is not enough left to complete the task. Examples of perpetual motion machines. Robert Boyle built a self-filling glass, one of the most intriguing experiments on the subject, as the machine works with certain liquids. The problem with this invention is that it only works with liquids that contain gases, and stops functioning when these gases dissipate. The drinking bird relies on the evaporation of water to keep its cycle going, and once the water in the glass runs out, it stops working. Even machines that rely on solar energy or a radioactive energy source cannot be considered perpetual motion machines, as that energy is also consumed. A possible solution arises: to reduce friction as much as possible by using magnetism, which would also provide a constant stimulus through electromagnetic attraction. This stimulus would be generated by the principle of attraction and repulsion of magnets. A stronger magnet induces the repulsion of the weaker magnet, which would be responsible for moving the wheel. And how do you keep the magnet spinning? A rotor would ensure that the magnet stays on its path. Still, several fixed magnets would be necessary for the free magnet to complete a full rotation. The theory seems to work, but in reality, it does not. This happens because, for the free magnet to continue on its path and complete the rotation, an attractive force equal to or greater than the repulsive force induced by the previous fixed magnet is needed. So far, it is impossible to create a perpetual motion machine because, even if infinite energy could be generated with magnetism to reduce friction, during the energy conversion to perform the task, it would disperse into other forms of energy. Even with the movement of atoms, energy is lost. In any case, the concepts of perpetual motion machines and infinite energy violate the principles of energy conservation and go against the laws of nature. Thank you for watching this video. If you enjoyed it, don't forget to like it and subscribe to my channel to receive more content like this. And tell me in the comments, what do you think about infinite energy? Tissue engineering and artificial organs. Welcome to our new video. In this video, I will present you some curiosities about the creation of artificial organs. The following aspects will be addressed: what is tissue engineering? How does the process of creating tissues work? Organs successfully replicated in the laboratory, clinical and laboratory examples, and advantages and disadvantages of artificial organs. Get ready to learn amazing things about organ engineering and surprise your friends with these interesting facts. What is tissue engineering? Tissue engineering is the science that is responsible for growing new cells to create tissues in the laboratory. To create these tissues, they mimic the nanostructure of body tissues. This science seeks to use biocompatible materials to avoid negative reactions in the organ. This material must serve as support, protect the cells that are around it, and enable cell regeneration. How does the process of creating fabrics work? For the reconstruction of organs, the use of various materials is investigated, such as artificial or natural ceramics, biodegradable plastics, and matrices of donated or animal-derived organs. In this last example, a decellularization process is executed to avoid immunological reactions. Decellularization consists of extracting the donor's cells and replacing them with those of the person who will receive the transplant. These materials serve as the basis for building the future organ and repeat cell signaling stimuli. This model is also loaded with cells from the patient, which speeds up the regeneration process. If there are not enough cells from the same tissue, stem cells or cells from other tissues are looked for. Previously, a sample of the cells of this organ is collected and these cells are cultured. These cells grown on top of the scaffold are seeded until they cover it. These cells need to be positioned in a specific place and the layers are placed one by one. There is also research that plans to use 3D printers loaded with biocompatible materials to speed up the process of creating this scaffold. The use of the 3D printer can reduce the creation time of the scaffold from 9 hours to a few minutes. Dr. Chijo's impression method was able to reduce this time to 9 minutes, however, it is still not capable of building complete organs. After this initial model preparation process, it is introduced into the bioreactor. This device is responsible for mimicking the functions of the organism to provide physiological conditions as close as possible to the human organism so that tissue development and maturation occur. The functions of the bioreactor are: control the temperature, exchange gases, nutrients and waste from the cells. The final tissue is implanted in the damaged organ to promote regeneration. Organs successfully replicated in the laboratory. In humans, until then it has been possible to implant flat organs, skin, tubes, blood vessels and urethra, and non-tubular hollow organs, stomach and bladder. Solid organs could not yet be implanted due to the complexity of these organs. One of the biggest problems in creating these organs is connecting the veins, arteries, and capillaries to supply blood and keep the organ alive. Clinical and laboratory examples. Molly Stevens is a UK professor who led research into the application of smart polymer systems for bone culture. This innovative method was able to create large amounts of mature human bone and other organs that are more difficult to replicate, such as the liver and pancreas. The best-known example of an artificial organ is the artificial skin that is generated from a collagen membrane, helping patients with burns or cosmetic surgery. Doris Taylor is a researcher from the United States who managed to decellularize the heart of a rat and inject heart cells. 8 days after the procedure, she stated that the heart was beating. Using the impression, Dr. Atala managed to build a miniature liver capable of metabolizing drugs. Now the next challenge is to make it life-size. An artificial ear was created that after 12 weeks of growth was implanted in a rat. The artificial organ looks similar to the natural ear and also retains its flexibility. This study was conducted by scientists at the Massachusetts General Hospital, Boston, and animal cells were used for this. Advantages and disadvantages of artificial organs. Spain is a reference in organ donations, yet the numbers of people who need a transplant exceed the number of donations. Tissue engineering could help heal this lack of organs. Another advantage that could be provided by lowering the price of artificial organ creation is the decrease in organ trafficking and kidnapping. Although there are already successful cases of artificial transplants in humans, more studies are still needed and the costs for this procedure are still very high. Since these organs are created with cells from the individual who is going to receive them, the risk of transplant rejection tends to decrease. Thanks for watching this video. If you liked it, don't forget to like and subscribe to my channel to receive more content of this type. And tell me in the comments, what is your favorite organ? Neuralink and Elon Musk's brain chip. Welcome to our new video. In this video, I'll introduce you to some curiosities about Elon Musk's brain chip. During the video, the following topics will be addressed: what is a chip, the Neuralink chip, the Synchron chip, laboratory studies, examples of other brain-computer devices. Get ready to learn amazing things about this device and surprise your friends with these interesting facts. Introduction to the Neuralink project. The main objective of the brain chip produced by Neuralink is to connect the human brain to the computer. Among the advantages of this implantation is the aid to the communication of people with paralysis, visual deficiency, and injuries. After implantation of the device, the patient will be able to control a computer with his mind. To do this, they need to understand brain impulses and convert them into digital results. Elon Musk stated that he intends to implement more functions to the device, such as driving a Tesla car with his mind, saving and reproducing memories, playing video games, and restoring locomotion to people who have had spinal cord damage. The American Agency Food and Drugs Administration, FDA, approved the clinical trials of the project. This represents a great achievement, since in order to execute a clinical test, several stages must be approved, such as preclinical tests, durability tests, and biocompatibility tests. Although the approval for clinical trials has been a great advance for this technology, it is important to highlight that it is still estimated that it will take 5 to 10 years for the commercialization of the device. For the implantation of the device, a perforation of the skull is required. The device is made up of intertwined wires and electrodes. Musk said that multiple chips can be implanted for different functions. What is a chip? Chips are devices that contain millions of transistors capable of transmitting information. The plates of these devices are made of semiconductor material, silicon, that incorporates an electronic circuit. Examples of devices with brain-computer interface. These devices translate neural stimulation into computer language to enable communication between the brain and electronic devices. Although the issue of implanting technological devices to potentiate or restore human capacities sounds like science fiction, there are already several people who use them. Did you know that approximately 200,000 people around the world already use such a device? The most common example is cochlear implants that allow deaf people to hear. There are devices that detect potential epileptic seizures and alert the patient to take preventive medication or avoid certain activities. You are probably wondering, but if a chip can do several functions and is activated by neural activity, how will it know what to do? Scientists normally analyze through electrodes, which are the most relevant neural activities when one executes these tasks. These characteristics are passed as vectors to the algorithms that will be in charge of interpreting the neural activities and selecting the desired action. Synchron is a company created in 2016 that, like Neuralink, is creating a brain chip to improve the quality of life of people with disabilities. Synchron has already been able to do its first implant in a living New York patient suffering from multiple sclerosis, a degenerative disease. The patient's jugular was used to insert the chip, and the procedure lasted about 2 hours. 48 hours after the procedure, the patient was already discharged. Synchron's chip is called a stent rod and, unlike Neuralink's, it is implanted intravenously, thus the company avoids surgical procedures that directly affect the brain. On the other hand, stent rod is larger than the Link chip. After approval for clinical trials, 4 more chips have already been implanted in Australian patients. So far no side effects have been reported. According to Synchron reports, 2 of the patients have managed to control a computer with their minds and perform some tasks. The chip. The previous prototype was connected to a device that went behind the ear and would be connected wirelessly to the chip. The previous chip also had very thin wires. Now the second device is no longer necessary, only the chip. The dimensions of the chip are currently 23 mm by 8 mm. Thanks to its small dimensions, it manages to go unnoticed, as it is covered by hair. Chip implantation does not require general anesthesia and can be done by a surgical robot. The patient can put it on and take it off. The idea is that the battery of the device is recharged at night. The communication between the chip and the computer or telephone is given via Bluetooth. Laboratory studies. Elon Musk showed 3 pigs, one with the chip implanted, another that received the chip and it was removed, and another virgin that never had the chip. The objective was to show that all pigs were healthy regardless of surgery or receipt of the device. In the demonstration, Gertrude's brain activity could be observed while she walked and sniffed. Musk claimed that Gertrude's device was plugged into her snout, and when she touched something with her snout, 1,000 electrodes were activated by her neural activities. He also did tests with rats and monkeys. In the latter case, he got the monkey to control a computer with her mind. Pager, the monkey, started playing ping pong with a controller to understand how the game works and to map the neural activities stimulated during the game. After the implantation of the chip, she continued playing ping pong, but now without a controller, only with her mind. Possible negative effects. It is thought that some people who use these implants could become highly dependent on these devices or have their sense of self altered. Although no negative effect has yet been reported in patients who have received the Synchron chip. The Neuralink chip has not yet been implanted in humans, so we do not know if it has any risk. Conclusion. The chip-type brain-computer interfaces are new technologies that still need more studies to assess their effects on humans, but that will present a before and after in the history of humanity, once they will allow the rehabilitation of people with disabilities and their better integration into society. This technology will also be revolutionary for people without disabilities, as it will increase efficiency and provide improvements in different areas. Thanks for watching this video. If you liked it, don't forget to like and subscribe to my channel to receive more content of this type. And tell me in the comments, which of these chips features do you find most interesting? Curious facts about the Egyptian mummies. Welcome to our new video. In this video, I'll present some curiosities about Egyptian mummies. Get ready to learn surprising things about this part of Egypt's history and amaze your friends with these interesting facts. Mummification in ancient Egypt aimed to prevent the natural process of decay. Mummification was crucial because of its religious significance, as the belief in the afterlife in Egyptian culture was linked to the preservation of the body. It consisted of a complex funerary ritual involving the gods Anubis, Nephthys, Aset, and Osiris. Death marked the beginning of a new cycle and the separation between the soul, Ba, and the body, Ka. Initially, the most used techniques were linen wraps or resin or natron shrouds. The legend of Osiris had a significant influence on the development of embalming techniques. In this legend, Anubis resurrects Osiris with the help of magical formulas. Hence, Osiris represents not only death, but also the rebirth of life, while Anubis is considered the patron of embalmers. Organs were extracted from the bodies, cleansed, embalmed, wrapped in linen cloths, and placed in 4 canopic jars representing the sons of Horus and protecting the organs: Amset, vessel with a human head guarding the liver; Hapi, vessel with a baboon head guarding the lungs; Kabasanuf, vessel with a falcon head, where the intestines were deposited; and Duamutef, vessel with a jackal head guarding the stomach. The heart was usually left inside the body because it symbolized the individuality of each person, being the seat of thought, feelings, life, and consciousness. Mummification reached its peak in the New Kingdom, 1550 BC, 1070 BC, managing to maintain the expression of the deceased's face. Examples of mummies from this period include Seti I and Ramses II. Did you know that the mummification process lasted for 70 days? The corpse was washed, the viscera removed, then the body was left to dry in the sun and covered with several layers of oils, ointments, aromatic oils, and conifer resin. In addition to the oils, beeswax was also applied to the body, which, thanks to its antibacterial and hydrophobic properties, played an important role in the embalming process. Once the previous stages were completed, the bandages were applied. Among the different layers of bandages wrapping the body, protective amulets were placed. In some cases, a network of ceramic beads was also applied. The body of the deceased was then placed in one or more sarcophagi. Sometimes a gold or painted mask covering the face of the deceased was placed. There were several embalming techniques, some more costly than others. During the Old and Middle Kingdoms, mummification was exclusively for members of the pharaoh's family and officials and courtiers whom the pharaoh granted this privilege. As mummification began to spread, workshops dedicated to embalming emerged. During this ritual, several people were involved, with the most prominent being the Lord of the Secrets, who wore an Anubis mask and was responsible for treating the deceased's head. The priestly readers would recite the ritual instructions and magical incantations. The cutters were responsible for making incisions and extracting the viscera from the corpses. A curious and somewhat peculiar fact is that mummy extract was prescribed as medicine during the Middle Ages. The name of this exquisite medicine was mumia, and it was indicated for the treatment of headaches, bubonic plague, and indigestion. It is thought that this medicine may have arisen from a mistranslation of Persian or Arabic texts. Different forms of mummy extract were found, often represented by the liquid released by dead bodies mixed with aloes, pieces of mummy flesh, or a spice found in the tombs of the dead. Initially, powdered human remains, resins, and pieces of mummies sold in European pharmacies were imported from Egypt, but even though authorities prohibited such behavior, trading with local remains continued. Thank you for watching this video. If you liked it, don't forget to like and subscribe to my channel for more content like this. And tell me in the comments, what do you think of mummy extract? How does cloning work? Have humans already been cloned? Welcome to our new video. In this video, I will present to you some curiosities about cloning. The following topics will be addressed: